Our next speaker will be Richard Burke.
Okay, good afternoon. Thank you for the invitation to speak today. I'll be presenting work by a PhD student of mine, Sebastian Judd-Moll. He's just finished up in the lab. In fact, yesterday was his last day, and he's off overseas to travel and not think about chloride channels for 6 months. So, the next slide, please. We're actually a heavy metals lab. We're interested in copper and zinc homeostasis, and we first became interested in this family of chloride channels, the voltagegated chloride channels, thinking that they may be involved in copper transport. We soon showed that that wasn't the case, but we decided they were interesting in their own respect. They typically form homodimers, and I just point out there are lots of chloride channels around, and we're not talking about all these ones down here. And these voltagegated chloride channels have been implicated in a number of functions, and in particular for today's talk, focus on the acidification of intracellular organelles. So here's a phylogeny with the human proteins and the Drosophila proteins in green. And you can see there are 3 clear clades. Up here we have, I'm going to call these cliques today, even though they're not actually cliques. So we have clique A here, and 4 mammalian homologs. Clique C is orthologous to clique 3, 4, and 5, and clique B is homologous to clique 6 and 7. And I'm going to focus on these 2 bottom clades here, clique B and clique C, and they both encode proteins that are thought to be antiporters, so they transport chloride in one direction and protons in the other direction. And mutations in some of these transporters result in human disease. So clique 5 mutations cause dense disease, which is a renal disease with low molecular weight proteinuria, and clique 7 mutants cause osteopetrosis, which is the opposite of osteoporosis. It's a dense, brittle bone disease, and these patients also show symptoms of neuronal thyroid lipofuscinosis, which is a subtype of lysosomal storage disorder. So we thought it would be interesting to look at the fly orthologs of these genes and find out more about the detailed cellular function. First of all, we looked at expression, and this is just the clique B data showing that it's expressed ubiquitously, and clique C is also expressed very widely in the fly. Looking at the localization of the 2 proteins, over on the left we have, in the top row, salivary gland cells expressing a clique B GFP fusion protein, and you can see the punctate intracellular pattern. Compare that to clique C, which is a very strong apical or luminal membrane staining here. There's also some on the basolateral membranes, and there's also some intracellular signal. If we look at that in more detail, we had some clues from the mammalian literature about where to look, where these proteins might be, and we see that clique B co-localizes nicely with lysa tracker. So you can see over here in the merge there are plenty of these puncta that are yellow, and they're indicating that clique B is on the either late endosome or lysosomal membranes. Compare that with clique C GFP, where, as well as this outer membrane staining, we also see some co-localization with RAB5, indicating that at least some of this protein is at the endosome. So moving over to the functional characterization of these 2 genes, and Sebastian generated null mutations of both of them by imprecise p-element excision. The clique B null mutant is homozygous viable. And down on the bottom left here, they have some hyperpigmentation defects, and we also see melanotic masses in the tumours, but otherwise they're relatively healthy. We can replicate this with ubiquitous knockdown using RNAi, and we can rescue that phenotype with a genomic rescue construct. Looking to see if there are any more phenotypes, and we found that they have a lifespan defect. So here are the mutants. They have a significantly truncated lifespan compared to their heterozygous siblings, and we can partially rescue that defect with our genomic rescue construct. Now, remember the clique B is homologous to clique 6 and 7, and mouse knockouts of those 2 genes show symptoms of neurodegeneration, neuronal thyroid lipofuscinosis. So we thought there might be some neurodegradation phenotypes in the fly, and we did the classic negative geotaxis knockdown assay, look at locomotor activity, and here we have 2 wild-type strains and the mutant over on the right here. And in young flies, the mutants are doing perfectly well. They show the same sort of locomotor activity as the controls. But if we look at older flies, so around 24 to 27 days old, and this is a couple of days before they really start to die off in that previous lifespan assay, we can see they're severely defective in locomotor activity. One interesting aspect about neuronal thyroid lipofuscinosis is there's been a strong link with heavy metal dyshomeostasis. And being a metals lab, we thought we'd have a look into that in more detail. In particular, zinc dyshomeostasis has been associated with NCLs, and the lysosome is a key organelle for the storage and detoxification of zinc, and cells actually remove excess zinc by lysosomal exocytosis. So what we've done here is compare wild type and mutant flies and look at their survival from early larval stages to adulthood on either normal food or food supplemented with concentrations of copper sulfate, zinc chloride, or iron chloride. And I've used here what I call sublethal concentrations, so they'll typically reduce viability by about 10 or 20% in a wild type fly. And hopefully what you can see here is that the mutants are on their own on normal food, have a slightly reduced survival, but this reduced survival is greatly exacerbated on the metals. In particular, zinc chloride has a very strong detrimental effect on the survival. So we think that these flies may have issues with dealing with heavy metal stress as well. Moving on to the CLC mutants, homozygotes in this case were larval lethal, so they died during the larval stages, and Baz has tracked the growth of these mutants compared to their heterozygous siblings, and you can see over 3 days they're slower growing, then they stop growing and they plateau out, and they remain. We think they make it to third instar, but they're very small third instar, as you can see over on the right here, and they eventually die by about 8 days, and they never pupate. And again, we can rescue this lethality with a genomic rescue construct. So since it was adult larval lethal, we wanted to do mosaic analysis to see what the cellular phenotype was. We never saw surviving clones in the adult tissue, so we turned to the imaginal discs. And here we're looking at late third instar imaginal discs, and we're inducing clones. They're GFP negative mutant clones, and the twin spots are GFP double positive. And you can see at 48 hours post-clone induction, so we induce clones at various early time points and then look at late third instar. At 48 hours, the clones are still surviving, no worries. 72 hours, we only see the wild-type twin spots in the wing pouch, but we do see some CLC mutant clones still in the hinge and the nodal regions, and by 96 hours, we only ever see the wild-type twin spots. We no longer see any mutant cells, and we can rescue mutant clone growth with our genomic rescue construct. So clearly these CLC mutant cells have a survival or proliferation disadvantage, and they've been outcompeted by the wild type cells, neighbouring cells. And this is just a quantification of that data. We wanted to see if they were in fact dying, and it was tunnel assays that have been carried out both at 2 days and 4 days post-clone induction. We don't see any tunnel staining at 2 days post-clone induction. We start to see some of the clones being tunnel positive at 3 days, but the very few remaining clones at 4 days are predominantly all tunnel positive, as we can see in this case over here. So yes, they are undergoing apoptosis as they've been outcompeted from the epithelia. So what's going wrong in these mutant cells? Well, we know from the mammalian literature that these proteins are thought to have a role in intracellular acidification, organelle acidification, so we thought we'd look at lysotracker to see if we see any defects in lysotracker. In the wing disc, you get very low levels of endogenous lysotracker signal, so I was a bit worried that we wouldn't be able to detect any loss of lysotracker. Luckily, what we did see in these mutant clones was an induction or an increase in the intensity of lysotracker signal. So here's an example of a 3-day-old clone, and here are the mutant cells, and you see a massive increase in the intensity of lysotracker. And again, this has been quantified over here on the right. Not exactly what we expected. We were expecting, if anything, to see a reduction in lysotracker signal. And this is just to show the quantification of lysotracker versus tunnel, and just to show you that at 2 days, we see about a third of the clones that are lysotracker positive or induced lysotracker, whereas none of them are tunnel positive. At 3 days, more of them are still lysotracker positive than tunnel. So the increased lysotrack signal is preceding the cell death or the tunnel signal, if you like, and may well be a cause of the death of these cells. So I'm doing okay for time, so I will actually show this slide. We also wanted to do some RNAi to see if there are any other phenotypes we could pick up. I suspect the CLC RNAi line is not very potent because given the mutant phenotypes we've seen, we didn't see much. One thing we did see, when we really pump up the system, 2 copies of the RNAi line at 29 degrees, after about 10 days or so, we see this interesting loss of bristle phenotype. This is using Panea-Gal4, which is expressed down the dorsal midline. And what's happening is the bristles are just snapping off at the base. So I call this a brittle bristle phenotype. So if anybody's ever seen this before in another mutant, please let me know afterwards. I'll be fascinated to know what this means. The other interesting thing here is that, again, knockdown of CLC-b or CLC-c, we don't really see much initially, but when we knock them both down together, we do see this loss of pigmentation phenotype. So I'm going to finish up there just by summarising what I've shown you today. We have 2 related but clearly different chloride proton antiporters or exchanges. CLC-b is localised to either the late endosome and or lysosome. The mutants are viable but have defects in lifespan, in locomotor activity, and in metal ion tolerance. And we don't think that there's actually a defect in lysosomal acidification here. It's never been detected in the mouse mutants, and we're looking at pretty subtle defects here, so it could be that the lysosome actually requires a certain content of chloride ions to function properly, and we're detecting defects in that. With CLC-c, we see it localised at the plasma membrane and at the early endosome, and instead of a decrease in organelle acidification, we actually see an increase in organelle acidification, and this protein, this gene, is clearly required for cell viability. So what we think might be happening here is that a failure of acidification of the early endosome actually blocks the recycling pathway back up to the plasma membrane, and that may be the cause of this increase, maybe increase in the size of the lysosomes as a response. So it just remains for me to thank Bas, who's over on the left there, for doing all the work, and you for your attention. I welcome any questions. Thank you.
I'll start. I'm curious if you have any ideas about which tissues require CLC-b function for the aging defect.
Which tissues require CLC-b? Yes. Just starting to do those experiments now. We're going to use RNAi knockdown to try and see if it's the central nervous system or the muscles that are actually responsible for those locomotor defects. Yeah. But at the moment, no ideas.
Did you look at any conditional stresses, such as starvation, for example, and see if maybe your lysosomal...
No, we haven't looked at any other conditional stresses. Bas had the idea that maybe the increase in lysotrack was actually a sign of autophagy in those dying cells, and he did look at the M-cherry-A to-G8 reporter, and he didn't see any signs of increased autophagy in that case.
Did I see on your first slide that it's a voltage-gated?
They're apparently voltage-gated. I'm not an expert in electrophysiology, but yes, they're well characterized as voltage-gated.
Is that relevant to any other functions? Like, how are they regulated in these other contexts?
I've struggled with that, and I really don't have any clues as to how voltage might feed into their role in cell proliferation or survival. I don't know.
Do you think the mutants are surviving so long because of a maternal supply, or do you think, if you look in S2 cells, if you take out the CLC, do they live?
It could be maternal supply. We haven't done germline clones, so I would have quite liked to have done that to see if we can remove that. It could also be, but even in the mitotic clones, they do survive for 2 or 3 days, so it could be that it's not absolutely essential, but eventually they have a proliferation survival defect just in comparison to the wild type cells, and they're unhappy and outcompeted. Okay. Thank you very much.